a visual reference as we talk about the process, it's a little bit easier. Okay? So, I've got a couple of videos. One is glycolysis. Okay? Because remember, we're going to talk about making ATP in relationship to the glucose molecule. So the first thing that happens is glycolysis. And then we have the cellular respiration. So if we can have a visual representation and we can refer back to it, I think it'll help. Okay?
Consumption step. Biological organisms require energy to survive. Glycolysis is one of the pathways cells use to transform sugars like glucose into biochemical energy in the form of ATP. In the cytosol of the cell, glycolysis converts glucose into pyruvate through a series of 10 enzymatic reactions. This process produces ATP, along with other products such as NADH that can be used later to produce even more ATP for the cell. Let's watch as these enzymes oxidize one glucose molecule into two pyruvate molecules. First, a kinase reaction adds a phosphate onto glucose to form glucose 6-phosphate. This is one of two energy consumption steps and is an irreversible reaction. Next, an isomerase reaction converts glucose 6-phosphate into fructose 6-phosphate by rearranging covalent bonds. Another kinase removes a phosphate group from ATP and gives it to fructose 6-phosphate to form fructose 1,6-bisphosphate. This is the second energy consumption step and an irreversible reaction. In the fourth step of glycolysis, a lyase reaction splits the 6-carbon fructose 1,6-bisphosphate into two 3-carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate is rearranged by another isomerase to form a second glyceraldehyde 3-phosphate. At this point in glycolysis, glucose has been metabolized into two glyceraldehyde 3-phosphates and two ATPs have been consumed. The next 5 steps of glycolysis are the energy-producing phase. In step 6, both glyceraldehyde 3-phosphates are oxidized to 1,3-bisphosphoglycerate by a dehydrogenase. This step produces one NADH for each oxidized glyceraldehyde 3-phosphate, for a total of 2 NADHs. These NADHs are later used to produce more ATP for the cell. In step 7, a kinase transfers a phosphate from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. This step is reversible even though ATP is formed. The next step involves a mutase reaction that moves the phosphate on the 3rd carbon of 3-phosphoglycerate to the 2nd carbon position to form 2-phosphoglycerate. In step 9, a lyase reaction removes water in 2-phosphoglycerate to form phosphoenol pyruvate. In the final step of glycolysis, a kinase reaction removes the phosphate group from phosphoenol pyruvate and donates it to ADP to form ATP and pyruvate. Like reactions 1 and 3, this step is irreversible. At this point, 2 pyruvate molecules, 4 ATPs, and 2 NADHs are formed for each glucose that was broken down in glycolysis.
So, at this point, this is glycolysis. Glyco referring to a glucose molecule, lyse meaning to break it. So that at the end, see these two pyruvates right here? Those have now, that's what we have gotten from that one glucose molecule. The other parts that we got, now this is glycolysis. This process only occurs in the cytoplasm. The benefit of having glycolysis take place, it can readily use glucose, and it will give me some energy. But it also produces lactic acid, which builds up in the cells and is not good. Okay? The reason for this process, before cells formed their relationship with mitochondria, this was the only way they got energy. So, if needed, cells of our body can carry out glycolysis to produce energy. It's not efficient, but we do it. For example, a marathon runner, once they have used their sources of energy, the way that their cells make the energy for the rest of the race, they make a lot of it by glycolysis. It's the reason that when they're done with the race, their muscles are very sore. Their muscles are sore because of the byproduct from glycolysis. So, glycolysis is a way for cells to make energy.
Would you say you know all of the 10 reactions that take place?
Oh, no. You don't need to know that either. I'm just trying to give you an idea that we have taken this glucose molecule, it went through all these reactions with all those proteins, okay, and it created energy. Not great, not efficient. We did create this right here, this NADH, which becomes important once we get cellular respiration. So, but at this point in time, you can make energy in just this way. Not efficient, but you can.
The pyruvates and NADHs could be used in aerobic respiration to produce more energy in the cell. Here, we depict glycolysis as a closed process. But in cells, substrates produced by other reactions can enter glycolysis at different points. For example, when an animal breaks down glycogen, glucose 6-phosphate is produced and can then enter the glycolysis pathway at the second step. Importantly, this means one less ATP is required for the pathway because the first ATP consuming step is skipped. Other sugars can also enter the glycolysis pathway at different points, each having a different effect on the net number of ATPs that are produced by glycolysis. These ATPs are important energy molecules required for many biochemical pathways and ultimately life itself. Glycolysis is a major contributor to the pool of ATP used in these pathways that are essential to the survival of biological organisms.
So the cells can make energy that way. Those two pyruvate and the NADH, if the cell does not need to, and it can carry out cellular respiration, that is more efficient. And it will use those
How do
products.
you turn that bite of food into a chemical that a
From
cell can recognize
glycolysis.
and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria.
Pay attention to anything that they deal with these little electron carriers. All right, pay attention to those.
And the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide.
So at this point, I've had 2 more electron carriers. I've created a product that now is going to move forward. And my byproduct at this point in time is now CO2. This is what has to go back out, make it to the lungs, and we breathe it out. Okay, CO2 is the byproduct of cellular respiration.
Were those the same two NADHs from?
No, these are 2 new ones.
So your
we've got
net's 4.
2 and 2 more.
Okay.
Mhm. And like I said, keep an eye, because you've got NADH and then you've got some FAD that's going to come in and be FADH. These terms stand for their names, all right? But what I want you to pay attention to is what is going to happen with these electrons.
The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate.
Did you notice another